Welcome, everyone. My name is David Regan, and I'm CEO of Sona Nanotech. With me today is our Chief Scientific Officer, Len Pagliaro. Our company is developing an innovative treatment for colorectal cancer that involves the injection of our uniquely biocompatible gold nanorods into tumors, and then the application of infrared light to heat them, eliminating tumors from the inside out. And we do this without harming the healthy tissue, as happens with radiation and chemo. It's effectively a superior photothermal therapy for cancer made possible by Sona's uniquely biocompatible gold nanorods. Today, we're going to go through and tell you about targeted hyperthermia therapy. And at the end of this presentation, we'll give you an update as to where we stand and the progress we've made in the last couple of months since we announced this back in March, following our merger with Seva Therapeutics. Of course, that merger came about following an interaction with Len, who came to us a year ago as a potential customer, saying that he could not find gold nanorods that were stable, biocompatible, and had scaled manufacturing. So we struck a supply agreement at that time. But the more we worked together, the more we realized that the gold nanorods were the critical element here, and in fact, likely the monetization mechanism of the therapy. And that if we came together, the two companies were much more likely to achieve approval faster and commercialization sooner. And so that's exactly what we did. Now, prior to actually formally coming together at Sona, we did our diligence on Len and his company. And in speaking with one particular surgical oncologist and asked about the merits of targeted hyperthermia therapy, this entire concept, he responded and said, "Look, David, let me put it to you this way. I just had to tell a 40-year-old patient that he had a rectal tumor. And when I told him that we just had to cut it out and there was no other option, he could not believe that that was it, that there were no other treatments available for him." It has such a dire impact on quality of life in terms of attaching a colostomy bag for the rest of his life. The clinician then went on to say, look, it's not a question if a more advanced type of selective and precision therapy for these awful types of cancers isn't going to happen. It's a question of when, given all the recent advances in nanotechnology and their medical applications. Len and I and our entire team, extended team, and our company are working to make that when happen now. So moving forward, of course, this is a preclinical development, early stages. So we'll seek safe harbor for the numerous forward-looking statements we make today. But it's something that we are extremely excited about and more encouraged day by day with the work we do to advance it, with things really looking much more interesting every piece that we put of the puzzle together. So a year and a half ago, we started evolving our team in order to bring the right team together to be able to develop a therapy based on the technology that we have in our gold nanorods. We're very fortunate to have a board that hits well above its weight for a company of our size, starting with the recently named chair, Mark Lavonin, who led Sanofi Pasteur to become a billion-dollar company, Walter Straps, who's CEO of Khosla Ventures Biotech. Neil Fraser is the longtime head of Medtronic Canada, of course, the Canadian arm of the Medtronic global juggernaut of medical device companies. Dr. Michael Gross, who's a surgeon and a corporate director, and of course, Jim McGann, who's a merchant banker, venture capitalist, and highly experienced in capital markets. We then brought together the project team. We knew this concept has real legs, so we needed to make sure we went at it with gusto and with the right capabilities. Brought on Glenn Canner, who's a specialist medical device product development consultant in the US, who's taken multiple medical device products from inception all the way to commercialization, which can be a really long road. Len, of course, who's the developer and originator of targeted hyperthermia, Colbert Singh, who's the co-inventor of our biocompatible gold nanorods, Sandra Brennan, head of our quality affairs, and myself on the commercial and project management side. We also have access to a group of tremendous experts, luminaries in this area. And we think, taken as a whole, this is the team that can take this technology to commercialization. Now, sadly, surgery is the cornerstone of curative therapy for rectal cancer in particular, which is always risky to do, especially for patients with comorbidities like the elderly, and can result in a very poor quality of life afterwards, as we mentioned. Now, physicians will refer to the tumor staging as good, bad, or ugly, depending on their advancement. But the reality is, in way too many cases, regardless of that initial assessment, they too often end in a total mesorectal excision. This is just one of the types of cancers we think we can have an impact on. And we're starting with this one because the alternatives are so limited and the outcomes of this type of surgery are so limiting on quality of life. We think this is where we can make the biggest difference fastest. Now, turn it over to Len to get into some of the past research that has been done.
Thanks very much, David. So we've done quite a bit of preclinical work doing studies at the bench level and then also on animals. And so this graph shows the result of a rodent model, a rodent tumor model, in which we tested 4 combinations. Now, the combination in the green light, in the green line that you see just says tumor shrinks. That is our photothermal therapy. That's the combination of injection of the gold nanorods, as David mentioned, and the infrared light that activates those gold nanorods. Very dramatic result. The other 3 lines in black and gray show combinations of the infrared light with saline or the gold nanorods without the infrared light and so forth. And so when we do these controlled experiments, we can see a dramatic difference when you're using the combination of photothermal therapy that we have developed. Here is an image from another study, a different study, images of a mouse. It's the same mouse in all images, a large multi-lobe tumor on the left. 19 days out, you can see the tumor mass is gone, a little bit of scabbing. 59 days out, the animal's healthy, running around. The CRO that did this study called it a durable cure, at least durable remission, I think would be a safe word for it, an excellent result. Now, 2/3 of the animals in this arm of the study were alive and healthy at 95 days after treatment. That's a very long time for these mouse studies. We eventually sacrificed the animals. We looked at their tissue histologically under a microscope, and there was no evidence of tumor. In contrast, with the drug of choice for this tumor model, all the animals were down by 28 days. So a significant improvement over drug of choice. Will we see that in all cases? Of course, we don't know that yet, but it's a very, very encouraging result at this stage of research. Now, here's how it works in practice in a human being. This is our concept. Of course, we have not been into humans yet, but we're going to use pretty much available technology, but it has been optimized for treatment in humans. So on the left is an image that's actually an electron micrograph, not a light micrograph image. And you can see a red blood cell. That's the red sphere, the red round blob in the picture. And then those little tiny specks inside it are nanorods. Those are gold nanorods. So to give you an idea, red blood cells are among the smallest cells in the body, and the nanorods are little specks in that red blood cell. So they're truly, truly, truly nano. They get pretty much everywhere in the body. However, they concentrate in tumors because tumors have a degree of leaky vasculature known as the enhanced permeability and retention effect. Now, our concept for delivering the infrared light to a region in the colon and the rectum is to use an existing instrument, and that would be a colonoscope. Colonoscopes have a channel through, put instruments through, and so we're actually working with a medical device developer to develop a device that will go through that free channel and deliver infrared light. And the image on the right shows the concept of what it would look like for a tumor inside the rectal region and infrared light being delivered by an endoscope.
Thanks, Len. So our plan going forward here is to build a mountain of data. It's the data on safety, biocompatibility, and efficacy that's required by not just regulators like the FDA and Health Canada, but by the market, and in order to be able to see exactly what it is we have and how well it works. The goal for us is to get into clinics as soon as possible, possibly next year. But what is paramount is that we do this properly. And we've taken in some tremendous third-party counsel from experts who have been extremely helpful in that regard. And one of the key learnings here we've had along the way is that there are something like 30% of medical devices that get cleared by the FDA that never get commercialized. And one of the lessons from that is you've got to, as indicated at the bottom of this slide here, engage with key stakeholders who are going to be the users, the affected parties, the payers, importantly, very early on in the process to make sure it's not just that your science is strong, but that you've got a product that's going to find a place in the market. Now, moving forward, I'm going to move fairly quickly through the next few slides because we're keen to get to the real updates for you as to of what we've accomplished in the last little while here. We talk about this different stages of colorectal cancer, rectal cancer in particular. We think we can apply THT, targeted hyperthermia therapy, to the certainly the first two, likely the third stage as well. And really the current standard of care there is the middle line, which tells you, a lot of treatments here that are quite distant in terms of the quality of care that's provided there relative to what we think can be provided with targeted hypothermia therapy. Len, do you want to talk about the secret sauce?
Sure. So when heat is used for cancer treatment these days, the overwhelming majority of treatments are what's called ablation, very high temperatures, above 55 degrees Celsius. And so if you're thinking about steak as an analogy for tissue, you're cooking the steak. It would be medium well to well done. What we're doing is very different from this. What we're doing is heating tissue to a gentle temperature of 44 degrees Celsius or 111 Fahrenheit. And that is quite different from cooking the tissue. If you use the steak analogy again, it would be warm. It's what's typically called blue rare, but it is not cooking the tissue. This is important for a number of reasons, primarily the fact that cancer cells are more heat sensitive than non-cancer cells. And so this temperature of 44 Celsius helps us select for cancer cells, and it does a number of good things in those tissues.
Yeah, and the current photothermal therapies that are out there really just blast energy and are indiscriminate in that way. Our concept will be more difficult in order to be able to maintain that temperature at a certain level at 44 degrees, as opposed to just blasting it and having all that collateral impact you see on the left there.
So there are at least 4 things we know about that targeted hyperthermia, this gentle temperature of 44 Celsius, 111 Fahrenheit can do. First of all, it shrinks tumors because tumors have different metabolism from non-tumor cells. It's known as the Warburg effect, and that's one of the reasons they're more heat sensitive. The second thing it does, and I think we need a button push, David. The second thing it does is to take out cancer stem cells, which tend to be heat sensitive, but they're also drug resistant, and so drugs don't work too well against them. The third thing that happens is that it increases perfusion to the tumors, and so if our therapy is used together with drugs, and likely it will be part of a combination therapy, then the drugs can potentially do their job better. And then last, but certainly not least, you get a degree of immune system stimulation because you get release of material, partially denatured antigens and cytokines and so on into circulation.
Thanks, Len. So let's go inside the lab now to see a quick demo of how an infrared light can pass non-thermal energy into a solution with gold nanorods that then convert that energy to heat. And here you see the first evolution of our light device, our infrared light device. You see it's turned on here with the purple light. This is little cuvette, has gold nanorods in it. The white item here is the thermometer probe. So you can start to watch the temperature rise fairly quickly. And we've got some more data later on we're going to share with you that's really interesting. And here we are in a fairly short order of time, temperatures up to that target 44 degrees or so. The light's now turned off, and that solution starts to drop in temperature. Again, it's not the light itself that's heating things up there. It's the gold nanorods that convert that non-thermal infrared energy into heat. And that's really important because this concept is about creating the heat that kills cancer cells from the inside out, not blasting the tumor with heat from the outside. Now, the Nanotechnology Characterization Lab of the US, which was set up by the National Cancer Institute and the FDA specifically to assist with medical developers of advanced technologies such as Sona Nanotech, by giving them access to sophisticated laboratory and personnel to be able to characterize and advance their technology, have done an extremely thorough process. It's a program that Len and his team got into. It's a prestigious and tough program to get into. They're very selective. It's a rare and valued resource, and so they don't open it up to just everyone. Nonetheless, this concept of targeted hypertherapy definitely merited it. And their assessments of us, multiple rounds of our gold nanorods, have been extremely positive. No endotoxins detected, no microbial contamination, all the other characterizations done of it. This is really helpful, as I mentioned, set up by the National Cancer Institute and the FDA. These folks, these scientists at the NCL went so far as to say, look, going forwards, if you want us to come and sit with you in your meetings with the FDA, we'd be happy to do that with you. That's something we're going to absolutely take them up on. Len.
So when you're taking a technology like this to market, there's a long path you have to follow, part of which is technology development, of course, part of which is the regulatory pathway. So things that have already been done in the field, already done or already improved, is first of all, using heat to treat cancer, a number of ways of doing that. As I mentioned, most are ablation. What we're doing is much better. It's hyperthermia. Photothermal treatment using infrared light devices. There is a version of that that's in the clinic in the US. It's not yet to market, but it's in the clinic, which is a big step. We think our version has numerous advantages over that one. The FDA has approved gold nanoparticles for injection, for systemic injection in humans in clinical trials. And then finally, a lot of efficacy and safety data in small animals. Now, what remains to be done? Well, there are a number of things, of course. And on the right, we can see, I think we need another button push here.
There you go.
That what we need to do, and we're working on currently, is delivering infrared light through an endoscope, and we'll have an update on that. Safety and efficacy in mammals and large mammals. Dosage of gold nanorods, what's the optimal dosing, the maximum tolerated dose, what's the optimized treatment time, and then last but certainly not least, intellectual property. We want to make sure we build a good intellectual property fence around our technology.
Yeah, so we really want to emphasize to folks who may not be familiar with some of the science and the regulatory aspects here that much of what we're talking about, there's been a tremendous amount of research done on it. Much of it has been done before. The things that we are focused on, the biggest challenges we have tend to be pretty operational, in fact. So obviously this is a compelling proposition regardless, from a quality of life perspective, from a science perspective, from a financial perspective, that flows. And we'll do a little fun with numbers here just to assure folks that there's a significant market available for a therapy like this. If you look at rectal cancer cases in the US alone, 46,000 new cases per year and rising, with the average age getting younger, by the way. We figure about 28,000 of those would be treatable by THT. So let's take an arbitrary, 10% of the market, if we're able to get that, and consider 2 applications at $20,000 per application for a revenue of $40,000 per patient. That alone is annual revenue of $112 million. Now extend that to colon, esophageal, and head and neck. With the same metrics, similar metrics, you're tallying up to some significant numbers here, US on a recurring annual basis. Len, I think you want to touch base?
Sure, and this is obviously a subjective assessment, but when we compare our treatment to the mainstays out there, surgery, drug therapy, radiation therapy, we think we have advantages in really every category. For efficacy, again, based on animal studies so far, we've seen excellent efficacy. We have an excellent safety profile, which is critically important, especially if we want to go in earlier for cancer. We have a great degree of precision, and that is very limited unwanted effects, both because of our ability to target the treatment and because of the fact that the gentle hyperthermia does not damage healthy tissues. And then last, but certainly not least these days, is affordability. We think in terms of reimbursement, we're going to have a very, very attractive option for payers.
And I'm just going to add in here a little bit that came out in a press release on July 27th that was just brought to my attention from the Biden administration as part of their moonshot cancer treatment approach that is apropos to one of the questions we just received in the chat box here. The quote from that press release is that surgical procedures are often the first treatment option for the approximately 2 million Americans diagnosed with cancer each year. However, current surgical technologies do not allow doctors to easily and fully distinguish cancer cells from normal surrounding tissue in the operating room. This can lead to repeat surgeries, a more difficult recovery, and cancer recurrence, as well as higher healthcare costs. We believe our treatment would address that directly because of the past of first of all, the prevalence of gold nanorods throughout the tissue and the casting area of the infrared light that could be applied, hitting not just the center of the tumor, but throughout, without any concern of them doing damage because it's being held at 44 degrees. Enough to kill cancer cells, not enough to kill healthy tissue.
So there are many ways of comparing our treatment to the competition, if you will. And of course, we've looked already in general at drug therapy, radiation therapy, and surgery. Now, if we zoom in a little bit and we looked at alternative nano approaches for cancer treatment, there are a couple of competitors that are on the radar. The first is Nanospectra Biosciences. They have a version of photothermal therapy that uses very, very large particles. They're round nanoshells that don't clear the body efficiently, and they use a laser. They're basically doing laser ablation. And so issues there with efficacy, safety, and clinical workflow, specialized equipment, specialized training required. Company called MagForce that uses magnetic nanoparticles for treating cancer. They have seen some reasonably good efficacy, but again, some issues with safety potentially. Clinical workflow and affordability, a big problem there because it requires specialized equipment, specialized training. And then finally, there are radiation approaches for cancer that use nanoparticles to enhance that radiation. These have some potential, but one of the things we're trying to do is to get away from radiation, ionizing radiation, to a safer means of treating the cancer.
And Len, there's a question here about whether the gold nanorods, once injected in the body, whether they stay in the body.
Yeah, that's a very good question. So again, based on small animal data and work that was done by the NCL program that David mentioned earlier, we see that the material is excreted in both the urine and the feces, which is a good thing from a safety perspective. It stays in the body for a time. The nanorods reach a maximal concentration in tumors 12 to 18 hours after injection. After that, they slowly wash out of the body. And so the half-time of clearance looks, again, from rodent models, like it's going to be about a week. So in a month, the vast majority of the material will be out of the body, that's a terrific thing from a safety perspective.
Yeah, and the FDA, of course, have had a nanoparticle protocol set from 2017 as to their expectations on that. Now, here we go. So we've got a mountain to climb ahead of us. This spring, we marshaled the team, the materials, and the plan, and we're pursuing it, and we have moved from base camp and are heading towards Camp 1 now. Very pleased with the progress to date. We've got what we think we need to conquer this mountain. Certainly, there are some steep pitches ahead and a tough long road, but we've assembled, as I say, the team, the right partners, and the right materials to be able to do something that's really, really worthwhile. Now, a little update as to where we are and where things have gone. And Len, do you want to speak to, we've sort of broken down our work into a couple of different streams of work. Certainly a critical one here is on the light device. Len, do you want to speak to from whence we've come?
Sure. So this is very exciting. We're working with our partner, Minitronics Medical, based in the Twin Cities area, on turning the concept from a bench device into the device that can actually go through an endoscope and into the human body. One of the requirements is to get the infrared light through a long fiber. The fiber is going to have to be about 1 meter long, which is quite long. This would be the length of a colonoscope that again goes into the lower GI tract and can be manipulated by a doctor. These instruments have a free channel that you can put tools through. Very often it's a snare type device. In our case, we're going to put a fiber that delivers infrared light. And this is proof of concept that's been done at Minitronics, showing, and you can see a graph on the lower red light, on the lower right, you can see a graph that shows heating, very, very efficient heating, and that's that red curve that goes up quite steeply. This is light being delivered through a fiber. The light source in this case is a laser. We've tested both laser and LED light sources in order to optimize them. The curve, the bluish curve that's below that is an LED light source, and then those two lower curves are what it looks like if you heat water, if you just have water in the cuvette instead of gold nanorods. So there's a clear difference, and clearly getting very, very efficient delivery of infrared light through the fiber, which is a critical first step in turning this from a bench curiosity into a real medical device.
And this is really the first time we saw that real strong delta between just the normal effect on water and what the difference is that the gold nanorods provide to this quick acceleration at heating. Len,
so we've done a number of things along the way as well. We've got a great experienced regulatory advisor on board. The lead is a former FDA person who has great experience and has been on the medical devices side, the CDRH and the FDA. We're in communications with the FDA. We are working on a submission and a pending pre-submission meeting with the FDA. This is part of the pathway toward applying for an investigational device exemption. And then we're looking at the strategy of having a 2-device system. One device would be the gold nanorods. The other device would be the light device, the infrared light device. That may be a more efficient route to market. On the right is a matrix. It's a bit complex, but it shows aspects of the medical device approval pathway. Now, injectable gold nanorods, although they're known to the FDA, are probably still going to be considered to be in a high-risk category. They have an excellent safety profile. What we believe will happen, and that's shown at number 1 there, what we believe will happen is that as we get closer to market, there will be predicates for this device. And because of that, in large part, and because of the excellent safety profile, they will be essentially downgraded to a medium risk, a moderate risk device, or a class 2 device. That's critically important because that means a shorter, less expensive regulatory path to market.
Great stuff. So the other 3 streams here of activity we have, the gold nanofrod manufacturing, obviously that's our core competence here. However, we've had to do a bunch of work in order to ramp up production in anticipation of preclinical trials and clinical trials, of course, preclinical trials being animal. And the fact that the FDA and Health Canada require something called GMP manufacturing standards, good manufacturing processes. So we've been working with a couple of potential partners in that area, doing diligence on them, enhancing our materials. We feel we're in a really strong position now. We like all the partners we're speaking with. We've got to just narrow it down, pick one of them, and announce that, and then proceed to work with them. On the preclinical trial plan, this is really important stuff. You've got to be able to articulate, and particularly with our meetings with the FDA, exactly what our plan is to prove to them the safety, to be able to get the data to prove to them the safety, biocompatibility, and to a lesser extent for them, the efficacy. Remember, their primary concern is that no harm gets done to humans, not so much around the efficacy elements of it, which is their focus. Nonetheless, we need that, and we are working with an academic group who are very keen and are developing a plan right now to do a study with our materials to prove and show the efficacy of this in animals. Finally, on the stakeholder front, we touched base on that earlier in talking about the importance of that. That's become, more and more apparent, particularly with our work with Metatronics on the light device, is the need to engage with the customer and stakeholders, buyers, other folks who have influence on it in the clinic, as to how it will be used and how best we can design the system to minimize the speed bumps and constraints on getting it into clinic. So, design it such that it meshes with their existing workflows, and you're not requiring, to the extent possible, new work, new procedures, etc. So, for instance, Len talked about getting our infrared light into a fiber that can be slid into the spare channel of an existing colonoscope or sigmoidoscope. That's critical. Make it easy for these guys, folks. Don't recreate the wheel if necessary. Likewise, on the community front. And sorry, let me just finish on the clinician side. So we're close to working with an organization that facilitates engagement from experts, so as to get that direct feedback from multiple colorectal surgeons and others who have a stake and are involved in that practice. Secondly, on the community engagement, we're doing outreach to 150 patient advocacy groups. This is Colorectal Cancer Canada, the Colorectal Cancer Alliance in the US, organizations all around the world whose followers and whose stakeholders are really interested to learn more about these types of innovative treatments that are on the horizon, and can really help us by giving it greater light to what it is we're doing. Now, Len, do you want to take us home?
Sure. Well, of course, one of our goals is to create good returns for investors. In addition to that, though, we think we can really have a big impact on cancer. And so when we look at the number of new cases and the number of deaths from the cancers that we believe we can address in the relatively near term, first target is rectal cancer. After that, more broadly, colon cancer, colorectal cancer, esophageal cancer, head and neck cancer. You can see that we're talking about, and again, US numbers here, 240,000 new cases every year and about 85,000 deaths. Can we eliminate all of those? Of course not. I don't think anything will, but we believe we can make a really big impact here because our treatment is extremely safe. It's extremely effective. It can be used early, and early detection is happening more and more for all cancer types. And finally, certainly not incidentally these days, it's very, very affordable, which is increasingly important.
Thanks, Len. Now, there are a couple of questions here in the box. I'm going to speak to some of them. Anyone else who's got questions, please feel free to hit that chat button and add them in here. One here is, if possible, when would the GNRs be considered for a potential vehicle for drug delivery? And could you do it at the same time as part of the initial therapy? Len, do you want to answer that one?
Sure, I can take a crack at it. There are a variety of nanoparticles that have been investigated for, and in some cases are being used for drug delivery of various kinds. Most of those are liposomal capsules that have a payload inside, if you will, that's essentially released inside of tumors. Nanorods could be used as a drug carrier. One of the reasons we did not go after that approach initially is that from a regulatory perspective, that then clearly becomes a drug device combination. And so the regulatory hurdles go up quite a bit. Once we see success with our current mode of therapy, which we believe we will, then certainly the door would be open to saying, okay, let's explore some drug device combination approaches to cancer that could perhaps be more effective or more targeted.
That's right. And certainly the concept of a drug delivery mechanism, lots of people working on that. There's been a lot of research done using nanoparticles. That tends to be, frankly, where most of the activity is, obviously driven by the biotechs and drug companies. Our focus is on the therapy. Another question here asking if we foresee any benefits realized following a North American patent being awarded. Has a lack of patent delayed further interest in other companies with projects, for instance, gold nanorod sales? No, I'd say the answer to that, no. We've got a patent that's been granted already in Korea, and it's pending in North America. We already have great interest in our gold nanorods, which there are some small sales of it. Here's what we think, though, is that with the continued work and validation by the NCL and the profile that targeted hyperthermia therapy is going to provide to Sona Nanotech, you're going to see much more inbound interest in folks wanting to work with our nanoparticles, our gold nanorods, especially given their unique status as the only gold nanorods that are made without the use of CTAB, the toxin that is used typically to elongate them. Any other questions today? We're always happy to take your questions. We've got a lot of work going on, but it's one of those types of projects that it's great work to be doing when you're making traction towards a really great outcome, and that's how we feel right now. Another question: how is the progress on the light device?
Well, I'd say it's excellent right now. We've had a number of steps in the development, working very closely with Minitronics. And so what we've got to do is, of course, take the version that was used on rodents and turn that into a version that can be used in humans, large animals. And I think we're well on the way to doing that. The demonstration that we can get light through a fiber and then deliver the energy required for heating at the other end of a very long fiber is excellent. That's very exciting. Are there still details to come? Of course there are, but that was really a critical step in terms of demonstrating proof of concept.
Right. We're focused here on de-risking matters as we go forward. So we want to find out very quickly, the extent to which the therapy works. So we don't want to take a lot of time and a lot of money to engineer the best possible light device. We're working quickly to get a prototype that we can use to validate and use in our efficacy studies and safety studies to show it. There'll then be another evolution, another iteration of the light device. We'll probably go through 3 or 4 of those before we actually get into the human trials. And of course, we'll learn something through each of those preclinical trials that will be then fed back into the development process and our work with Minitronics. Okay, I think I'm going to hold it at that. There's some other questions around milestones and whatnot, but I think we've given the guidance we've got here today on the path forward. Always happy to take calls with you guys, and thank you for tuning in today. Stay tuned. Lots going on. We're on the cusp of some great things we think happening, and close to some partners being appointed and getting to work in earnest on some of the studies in the fall. Thank you.